All right, everyone, if you are watching this video, what you're going to get is just a very quick run through of all of the organelles, how they work together, and a review of their functions, basically. So here we go. So before we even go into each of the organelles, let's just really quickly lay out these three things. First of all, nothing in the cell is going to work by itself. All of the organelles are dependent on the proper functioning of the organelles that it's around. So within the cell, you need all of the parts to work. They give us an example here. They're talking about a macrophage or a macrophage, and essentially all that is a cell that gobbles up foreign things. So in this case, macrophages are usually associated with white blood cells or the immune system. So in this example, they're saying that a macrophage ability to take care of a pathogen, to ingest or rather like engulf a bacteria and destroy it, there has to be a couple of things that are working properly in order for that to happen. And so if we think about the bacteria landing or being close to one of the cells, so it's in the extracellular matrix, we need for the plasma membrane or the cell membrane to work properly so that it can engulf it. Once that happens, it kind of like pinches off so that that bacterial cell is enclosed within a vesicle. And from there, we need that vesicle to move correctly along the cytoskeleton tract that's laid down. We need a lysosome coming from the Golgi body to intersect that bacteria ingested pathogen, and then we need those two to combine. So the vesicle that contains the bacteria and the lysosome itself have to kind of put their cell membranes together, and only then will the hydrolytic enzymes inside of the lysosome work. And so in order for us to be able to destroy the pathogen, if the cytoskeleton tract is messed up, if the ability of the macrophage to engulf the bacteria is not up to par, then we get sick and we can't cure ourselves, or our body cannot rid itself of the pathogen. So again, these parts are contingent on the one before it doing its job. And then finally, the third bullet point is just saying that there has to be order within a cell in order for the specific function and roles of all of these organelles to be carried out purposefully. So the first organelle that we're looking at is the nucleus. It's a great place to start. We're in the middle. It houses the genetic material. Everything that needs to happen within a cell will come from orders from the DNA that is housed within the nucleus. So when we look at the nucleus, we notice that there is DNA there. There's also something called a nucleolus, which holds the instructions for making ribosomes. Ribosomes are important in transcribing DNA. So in other words, the ribosomes are going to work to read specific portions of the genetic code, because it would be a waste of time to read the entire thing just to make one specific part or to have one specific thing happen. So anyway, yeah, the ribosome is going to slide along a portion of the DNA that needs to be copied, and from there it creates something called mRNA or messenger RNA. Messenger RNA leaves the nucleus through one of the nuclear pores, and then we go into the space that we call the endomembrane system. The first thing you would encounter once you leave through one of the nuclear pores is the ER. So the endoplasmic reticulum is there. We have smooth ER and we have rough ER. Let's really quickly cover rough ER. So rough ER, rough endoplasmic reticulum, is studded with ribosomes, and it gives it that bumpy characteristic that we see in different slides of endoplasmic reticulum. So what are the ribosomes doing on the ER? They're doing the same thing that a free ribosome would be doing. They are copying, or not copying, but they're going through with making the actual protein that may be coming from that mRNA code that has left through the nuclear pore. So these ribosomes are further reading it. You have something called tRNA, transfer RNA, and its job is to bring those amino acids that are supposed to be put together to form a polypeptide chain. So this is going to happen in the rough ER. If we're in the smooth ER, we're talking about making phospholipids, steroids. They essentially want to help with detox processes also. So anything that you would need that is lipid based would be coming from the smooth endoplasmic reticulum. Once we leave there, we're on a cytoskeleton track to go to the Golgi apparatus. And the way that we get from the ER to the Golgi is via a cytoskeleton track. So those microfilaments and microtubules and the intermediate filaments, they're going to be present and necessary to keep the cell, first of all, in the correct shape. Second of all, keep the vesicles moving on the correct track. Once the product that enters the Golgi on the cis side like goes through the layers to the trans side and it comes off of the Golgi, it's placed in another vesicle, and then that vesicle is set on maybe another track, and that track is gonna give rise to wherever the cell is, wherever the molecule is supposed to be going, or the vesicle that is housing the molecule is supposed to be going. So we have lysosomes followed by the Golgi apparatus. Remember, lysosomes can come off of the Golgi. In that white blood cell example, so the macrophage example, we need to make sure that the Golgi is working correctly to create the proper lysosome to destroy the bacteria. So from there, lysosomes have these hydrolytic enzymes. They are not functioning when they are by themselves. It is only when they combine with some external pathogen, or even it could be like maybe a carb or something that we take in from the ECM, the extracellular matrix, and we need to break down. So the hydrolytic enzymes go to work only after they're activated via the joining of this other vesicle. So that is how the lysosomes work. Let's talk about the vacuoles. So vacuoles are the things we're going to find inside of a plant cell. They can store water, so they could be a water vacuole. They can also house nutrients, food, so they can be a food vacuole. In the case of a plant that has some waste that needs to be discarded, but the cell's not exactly ready to flush itself, you can store waste there as well. So you can have a waste vacuole. And again, the function of that in a plant cell only is to house one of those three things that I just named. So digestion, storage, waste, disposal, water balance, plant cell growth, and protection. All right. Last one, these are the powerhouse organelles. So we have mitochondria. The job of the mitochondria is to do cellular respiration. When you breathe in oxygen, you eat food. The mitochondria is what is responsible for taking those two products that you've just ingested and inhaled, and then to make energy from them. Energy in the form of adenosine triphosphate, or ATP. That is the energy currency of the cell. So when we eat and when we breathe, the mitochondria are going to transfer that energy source into a usable form that could be stored for later use if necessary. So that's the mitochondria's job. If we were looking at a plant cell, we would look at chloroplasts. Chloroplasts also deal with energy making. So in this case, we're going to use photons from the sun, so light energy. Not always the sun. It could be also a lamp or some special bulb that gives the same light waves off, or wavelengths of light off. Chloroplast takes those, and so with the presence of the photon and then carbon dioxide, which is the waste product that comes from the mitochondria, it will be able to, and some H2O, you need water there too. It's able to do photosynthesis, and photosynthesis yields glucose, and that's the thing that we need to eat, and it also gives off a byproduct of oxygen, which we need to breathe. So these two organelles work synonymously. They're synchronized. Last one, peroxisomes. All you need to know is that they're sandwiched usually in between these energy organelles. That's why they're placed here together. They have some enzyme in them that allows them to remove hydrogens from one molecule and essentially put them on an oxygen molecule of another molecule. I don't know another word for it. So then what happens is when we do that process, you get hydrogen peroxide as a byproduct. By itself, it can be toxic, but luckily we have something called catalase, which is an enzyme that helps to break down the H2O2 and make it non-toxic. And that's it. 10 minutes, 24 seconds, basically 10 minutes and 30 seconds. All organelles covered. Done.